what we're going to do is talk about chemical synaptic transmission, and we'd started that last time. And where we finished at the end of the lecture on Friday was describing the experiments that were done in the early 1920s by a guy named Otto Loewi. And what Otto Loewi had done is he had recognized that an important idea in all of neurobiology was sorting out whether or not the transmission between the nervous system and its target would be through a chemical. And he sorted that problem out by isolating a frog heart with the vagus nerve attached and recognizing that if he stimulated the vagus nerve, that would release some kind of substance that effectively slowed down the heart. He then proved that it was a chemical substance simply by stimulating the heart in a bath, taking a tiny aliquot of the fluid from one bath, putting it in another bath with a heart, and the other heart slowed down. That indicated that the stimulation of the nerve, stimulation of the vagus nerve, led to the release of some kind of chemical that affected the heart rate. We called that molecule Vagusstoff. We now know that that molecule is acetylcholine, and he worked with a fellow named Henry Dale at Cambridge to sort that out. And together in 1936, they shared the Nobel Prize. And so this is a very important insight into how the nervous system functioned. What we're going to talk about today is we're going to start with what's going on in the presynaptic element, the axon terminal, the output region of one neuron, and talk about its structure. And we'll lay out some things here that are probably familiar to you. We're going to find that the output region of a neuron is filled full of synaptic vesicles, thousands of these synaptic vesicles. But that a subset of the vesicles is nestled up against the membrane at a particular site called active zones. And one of the major components of the active zones are transmembrane proteins called voltage-gated calcium channels. So at the active zones, we're going to have synaptic vesicles right next door to the plasma membrane, right beside the voltage-gated calcium channels. And what we'll see is that when an action potential arrives at this site, the arrival of the action potential at this site delivers positive charge. And that positive charge then delivered at that site opens the voltage-gated calcium channels. Recognize that what we'll have here is a tremendous gradient. The outside of the cell will have calcium concentrations in the range of 2 to 3 millimolar. The concentration of calcium inside the cell will be in terms of 100 nanomolar, 10th micromolar. So a tremendous gradient. That means when we open these voltage-gated calcium channels, calcium comes rushing in. And it was known for a long time that calcium was necessary to trigger the release of neurotransmitter. But it took many years to sort out the molecular biology of it. So we'll walk our way through the idea here first of the anatomy. We'll talk about the morphology of the vesicles, where these vesicles are clustered at the membrane, how the clustering at the membrane is associated with voltage-gated calcium channels, and then how a series of experiments, some done in yeast, some done from cell biology and vesicle cycling inside the cell, and some done from brains, came together to generate something known as the Snare hypothesis. In the Snare hypothesis, there are two major kinds of proteins, one relegated to the vesicle and one regulated to the presynaptic membrane or target. The proteins in the vesicles are called V-snares. And the one we'll talk about is vesicle-associated membrane protein. But there are a corresponding set of proteins found in the axon terminal and plasma membrane. And these are called target or T-snares. The two we'll talk about are syntaxin and SNAP25. This one is a transmembrane protein. This is a membrane-associated protein. The most important idea here, though, is that a component of the V-snares and a component of the T-snares are alpha helices in the cytoplasm. And then when the calcium sensor, known as synaptotagmin, binds calcium, the calcium that's come into the cell through the voltage-gated calcium channels, when this calcium sensor binds calcium, that triggers a winding together of the alpha helical regions of syntaxin, SNAP25, and VAMP. By winding together these alpha helical regions, we basically form a kind of knot in those proteins. And by forming a kind of knot, it literally pulls the synaptic vesicle phospholipid bilayer into the plasma membrane lipid bilayer, smushes the two together, and results in exocytosis. For years, they'd understood there was exocytosis. For years, they understood the calcium was necessary. But it wasn't until they sorted out the molecular biology that they could put the entire story together. And this Snare hypothesis won the Nobel Prize in about, I think it was 2012. But that tells the story of how we release neurotransmitter. It doesn't really tell us what the fate of the neurotransmitter is and how the next steps proceed. After all, what we're trying to think about here is the communication between the input region of one cell and the output region of another. The output story is the release of neurotransmitter. The input story is how a cell responds to the neurotransmitter. That's going to be the postsynaptic element. The postsynaptic element, or the input region of a neuron, contains a particular kind of protein, thousands or millions of these proteins, called ligand-gated ionotropic channels. Again, there's meaning in this term. We've mentioned them in passing, but these ligand-gated ionotropic channels, what this means is the ligand-gated ionotropic channel will bind a neurotransmitter. When the ligand-gated ionotropic channel binds a neurotransmitter, that leads to the opening of the channel, and that open channel allows for ions to move through the channel. So now we have a new way of triggering the opening of a channel, a new way of regulating the movement of ions across a membrane. We've talked about voltage-gated channels that are regulated by positive charge. We've talked about the stretch-activated channels that are regulated by the movement of the membrane. And now we're going to talk about a really new set of transmembrane proteins, the ligand-gated ionotropic channel. These are the channels that are operated by neurotransmitter. And in the first run-through, we're going to talk about these channels as being ones that become permeable to a cation, to sodium. If sodium is going to come into the cell, we know that that influx of sodium is going to produce a depolarization. It's a depolarization which we're going to now term an excitatory postsynaptic potential, or EPSP. Now, one of the ideas I want to drive home here is that the excitatory postsynaptic potential that's generated by the release of the molecules in an individual vesicle is precious small. In voltage terms, it's less than a millivolt, well less than a millivolt. It represents the opening of a few hundred to a few thousand ligand-gated ionotropic channels. Remember that whenever you see this term, ligand-gated ionotropic channel, put those proteins out on the input region of the cell. The voltage-gated channels are relegated to the conductor region, but these are found in the input region. So now we're going to use neurotransmitter to open these channels. When the channels open, they let just a little bit of charge through. This charge is so small that it only depolarizes the membrane at that site by a millivolt. You'll remember, though, that when we drew the pineapple neuron, that we said there was an input region and a conductor region. The input region is where we put these ligand-gated ionotropic channels. Where we put the voltage-gated channels, we relegated or isolated the voltage-gated channels to the conductal zone. So now we have to figure out a way where the excitatory postsynaptic potential produced by opening of ligand-gated channels leads to the opening of voltage-gated channels to start an action potential. And one of the ideas that will come through from this is that an individual EPSP produced by the neurotransmitter in an individual vesicle is woefully too small to bring the membrane at the trigger zone to threshold. That means we have to add these potentials together. Several hundred of these in some cases are needed to bring the membrane to threshold at the trigger zone, and we'll work our way through that. But let's start here with the structure of the presynaptic element. This is really the output region of a neuron. Well, in the 1950s and early 1960s, when they first began use electron microscopy to look at biological specimens, one of the things they found whenever they would cut through an axon terminal, they found that the cytoplasm in this region was chock-a-block full of these spherical structures, which they termed synaptic vesicles. But they recognized that there appeared to be two families of these synaptic vesicles. One of them was set back here in the cytoplasm, but there was a subset that was nestled right up against the membrane. And when they looked more closely, they found that where these vesicles were nestled up against the membrane, they also found in those corresponding regions, they found voltage-gated calcium channels. Taken together, the sites where the vesicles were nestled against the membrane, along with the calcium channels, they named this region the so-called active zone. The reason they called it an active zone is they did a series of experiments and they described how the vesicular membrane would become one with the plasma membrane such that there was neurotransmitter release. Via exocytosis. Now, the morphologists and the physiologists were working together on this problem, and basically the electrophysiologists said that the axon terminal depolarized. They said that the axon depolarized when the action potential arrived. Well, that's just a long-winded way of saying that when the voltage-gated channels along the conductal region would deliver an action potential at the axon terminal, they would deliver positive charge in the form of sodium. Well, although they recognized that the sodium was necessary to depolarize the membrane, they also began to recognize that depolarization in itself was insufficient. The depolarization itself was insufficient to cause neurotransmitter release. They recognized from these experiments that what was required was extracellular calcium. If they would do the experiment on synapses where there was no extracellular calcium, where they had removed the extracellular calcium, they could show that they could depolarize the axon terminal. They could get an action potential to arrive. But without extracellular calcium, no transmitter was released. So in concert with the morphologists, they then began to recognize that what was happening here is that calcium, which in the extracellular space, extracellular calcium was in the neighborhood of about 2 to 3 millimolar. That the concentration of calcium on the inside of the cell, the cytoplasmic concentration of calcium, was in the neighborhood of about 0.1 micromolar. So like a 20,000-fold difference between the calcium concentration outside to inside. They then began to see that what was going on here was that when the action potential arrived and delivered a depolarization, that delivered positive charge, and that these voltage-gated calcium channels responded to that positive charge by opening their gates. When those gates opened, calcium came in. And they then recognized that the influx of calcium would lead to the fusion of the synaptic vesicle membrane with the plasma membrane. And this process of exocytosis typically released about 5,000 to at most 10,000 molecules of neurotransmitter. This is another way of saying that each vesicle contains about 5,000 to 10,000 neurotransmitter molecules. So we jam-pack about 10,000 neurotransmitter molecules into this sphere that's no more than about 50 nanometers in diameter. So the concentration of acetylcholine, or concentration of neurotransmitter, is pretty high inside these cells. Now at this point, they recognized calcium was necessary to trigger release. They recognized that they needed a depolarization to open the voltage-gated calcium channels, and that there were a subset of vesicles clustered right against the membrane at the site of the calcium channels. And the electrophysiologist, the morphologist, then envisioned this simply as a calcium in some way allowing for the synaptic vesicle to get so close to the plasma membrane that magically the two became one. The problem with that issue was that description, that story, was that the physical chemists came along and sort of threw cold water on the whole thing, because the physical chemists said, we know that there's a phospholipid bilayer. We know there's a phospholipid bilayer from the vesicle, and we know that there's a phospholipid bilayer of the plasma membrane. And the physical chemists basically said, we know that these phospholipid heads have positive charges on them. And they said to the morphologists and electrophysiologists, you tell me how you're going to bring these positive charges this close together and not have them repel. It's physically impossible. You have a real electrostatic problem. So this sent the physiologists basically and the morphologists back to the drawing board and invoked now a set of experiments that were done primarily by molecular biologists. So the initial work was physiological and morphological, and then there were a group of molecular biologists who in fact got the Nobel Prize in 2012 for this next story. The next story is said to be the Snare hypothesis. And the Snare hypothesis holds that there are vesicular proteins called V-snares and target proteins called T-snares. And the Snare hypothesis says that these proteins entangle to fuse the vesicular and plasma membranes. I won't go through all the details here, but I think it's an interesting sort of story in the coming together of a number of different lines of research. There was one guy, Randy Schekman, who was working on yeast. And he recognized that there were secretion mutants. And there was another fellow working on named James Rothman, and he was interested in vesicle trafficking. So he was interested in how vesicles were being targeted from the ER to the Golgi and then out to the plasma membrane, which we've talked about. And then finally, there was the question of neurotransmitter release in the brain. Tom Suthoff, who was interested in neurotransmitter release in the brain, had been isolating synaptosomes. He'd been basically grinding up brains and isolating the pre- and postsynaptic cells. And when he did that, he isolated a series of vesicular and membrane proteins. It then became apparent that there was a kind of conservation among all these proteins, that the secretion mutants all the way back into single-celled yeast had corresponding proteins that were important in allowing vesicles to fuse, allowing vesicles from the endoplasmic reticulum to fuse with the membranes of the Golgi apparatus, and finally to go out to the surface and fuse the plasma membrane. And then those very same proteins, or their cousins, were now being found at the so-called synaptosomes, the enriched regions of the brain where there are axon terminals and output regions of one set of cells and input regions of another set of cells. In the end, they found that the following story was consistent. And what I want to do up here on the right-hand side is I want to take the image that's shown here and enlarge that and put together the relationship of calcium influx, V snares and T snares, into the Snare hypothesis for transmitter release. So we're going to talk about over here is the Snare hypothesis for neurotransmitter release. So this is meant to represent the synaptic vesicle. This will be the synaptic vesicle at the plasma membrane. We could even say these are the synaptic vesicles docked at the trigger zone. So from what we've described, the plasma membrane here and the vesicular membrane become precious close to one another, and they're flanked at these sites by the transmembrane proteins we've described as the voltage-gated calcium channels. Now, on the synaptic vesicle, there's a transmembrane protein. So it comes across the plasma membrane. Then it has this alpha helical region out here in the cytoplasm. This is the alpha helical region of a protein called vesicle associated membrane protein. You'll often see this by an alternative name called synaptobrevin. Whether we call it synaptobrevin or whether we call it vesicle-associated membrane protein, it's a transmembrane protein found in the vesicle. Then at the at the active zones, there will be then a transmembrane protein I'm going to put here in red. It also has this alpha helical region. The name of this one is called syntaxin, Syntaxin. And syntaxin is also a transmembrane protein. The third character in this play, I'll put it in green. It has actually 2 alpha helical regions to it. This molecule's name is SNAP 25. Where VAMP and syntaxin are both transmembrane proteins, this was a little different. It's a little different in that it's attached covalently to the plasma membrane by fatty acids. It's pamidylated. That's a detail. But the SNAP25 and the syntaxin, SNAP25 and syntaxin, taken together, these guys are said to be the target snares. These are the T snares. Whereas the VAMP, the vesicle-associated membrane protein, or synaptobrevin, is a V snare. One more character in here, and I'll put this one in kind of brown up here, is also a transmembrane protein. It's also in the vesicle. I'll put a little tag on it here. And this molecule is synaptotagmin. Synaptotagmin is a really important protein in this story because synaptotagmin is the calcium sensor. It's in fact a calcium binding protein. So what we're going to do here is we're going to try to follow through what we have over here and now explain it in molecular terms. And what we see again is that when the action potential arrives, the voltage-gated calcium channels open and the calcium comes in. When the calcium binds to the synaptotagmin in ways that we aren't quite sure of yet, the binding of the calcium to the synaptotagmin leads to the purple VAMP, the green SNAP25, and the red syntaxin to wind together. They wind one around the other. So the alpha helices now form a coiled structure. When they form this coiled structure, when they wind around one another, they literally reduce the distance between the phospholipid bilayer of the vesicle and the phospholipid bilayer of the plasma membrane. They literally smush these two together. They bring them so close together that they overcome that energy of repulsion we talked about in the upper board. Mechanically, they simply overcome that energy of repulsion. They force these guys close together, and the phospholipid bilayer of the vesicle becomes one with the phospholipid bilayer of the plasma membrane, and we get the process of exocytosis. When this vesicle now forms the plasma membrane, the neurotransmitter inside the vesicle, the 5,000 to 10,000 molecules of neurotransmitter, are released to the outside world. Now, it's a detail here, but it's interesting to me because we still don't have this completely sorted out. There are two models here. One model is that when the calcium binds to the synaptotagmin, that's somehow instructive. What we mean by that is the binding of the calcium to the synaptotagmin in some way tells these V snares and T snares to wind together and smush the phospholipid bilayers together. But there's another model, and the one that's actually has favor now is one where under normal low calcium conditions, the synaptotagmin is preventing these alpha helical regions of winding together. And that when the calcium binds the synaptotagmin, the change in charge structure with binding all the calcium to this site now allows it to let go of the alpha helical regions, and the default pathway is for these guys to wind together and smush the vesicular phospholipid bilayer into the phospholipid bilayer of the plasma membrane. Both of those possibilities are still being entertained. The one, as far as I know, that has predominance right now is that under normal circumstances, the synaptotagmin is in the way, And then when we bind calcium, it allows for the default pathway of these guys winding together. Yes?
So is this like acting on bringing the vesicles towards that, or do the vesicles just kind of like take it away?
Okay, this is a great question. The question here, which I have blithely ignored, is how do we manage to get these vesicles, which are back here in the so-called reserve pool, up close to the membrane? We know that in part that these vesicles are tied into the cytoskeleton. They're tied in by a molecule called synapsin. And if we phosphorylate that synapsin, the synapsin will let go of the vesicle. So it's now no longer tied into the cytoskeleton. We think then the myosin takes over and starts walking the vesicle closer to the membrane. But there are a whole bunch of other molecules in here, UNC13 and a couple of small GTP binding proteins, not the trimeric ones, but some other ones, RAS and RAC and Rho. All of those seem to be involved in some way that, as far as I know, are not clear, of bringing this vesicle right into this spot and orienting it beside the calcium channels with respect to these V snares and T snares. But you're absolutely right. There's a whole round of research that's still going on to try to understand how we move these vesicles from the reserve pool into what we call the readily releasable pool. And there's another round of arguments that are going on that says that many of these vesicles, when they're recycled, which we haven't even talked about, that what we'll do is we'll reclaim this membrane and reload the vesicles to the neurotransmitter, that appears to, and many people say that's a local event. And so that's happening right here. Last in, last neurotransmitter and first out. Whereas others will get caught up in the reserve pool, and we only use those occasionally. So you're absolutely right. There's a whole round of research here trying to think about what the sequence of events are and the molecules that are involved to bring the vesicles from one place to another and put them into the right spot and how they're targeted properly. That, as far as I know, is not completely worked out. But you're right, it's an important aspect of this whole story. But what I want you guys to know for this story is the T-snare, V-snare story and the release of the neurotransmitter. There and then I'll go there.
Is that extracellular calcium the same as the receptor substance?
Oh, so the question is, we were talking about receptor substance. This is the Langley story. And the Langley story that I want you to emphasize is the receptor substance being the ligand-gated inotropic channel we're going to talk about here in a moment. That what Langley was seeing when he spritzed on nicotine was he was seeing the binding of the nicotine to what we now know as a nicotinic acetylcholine receptor. And the binding of the nicotine opened the nicotinic acetylcholine receptor. And only decades after that did we sort out, particularly with the neuromuscular synapse, that nicotinic acetylcholine receptor was binding to acetylcholine, being released by vesicles to the neuromuscular synapse. And that story was a long story, sorting out the structure of the acetylcholine receptor. And then a question in the back.
Yeah, whoever the gal was, I was thinking along the same lines, like, great thinker there. The question, though, in that, right, I'm not going to try to take it too far to keep my mouth shut, but there has to be some stolen real estate for those voltage-gated channels, right?
There is some stolen real estate for the voltage-gated channels.
It kind of flows like a river, mosaic. You see the phospholipid bilayer shifting around those. Are those voltage-gated channels anchored?
They're anchored. The voltage-gated channels are anchored, and one of the first indications that these voltage-gated channels were associated with transmitter release was in a morphological view called freeze fracture. And what they had done is they'd broken open the phospholipid bilayer, shadowed that phospholipid bilayer with platinum, and looked at those structures. And wherever they found exocytotic pits, they found associated with them these particles. We now are pretty sure those particles represent the voltage-gated calcium channels. But there was a morphological correlation between the sites where the neurotransmitter is being released by exocytosis and those bumps in the membrane. So they saw those, and those are anchored. Those are anchored into the cytoplasm. Yes?
What kind of force is required to overcome the electrostatic repulsion between the two phospholipid bilayers?
So the question here is, what is the force that's overcoming the electrostatic repulsion between the two phospholipid bilayers? That's this repulsive force. What we think is going on here is we overcome that electrostatic repulsion force by winding together these alpha helices.
But how much force are we talking?
Someone knows, I don't know.
Okay.
These things have been measured. And you can calculate the electrostatic forces, and I think you can calculate the forces generated by the winding together of the alpha helices. Now, okay, the truth of the matter is once these are formed together, that seems to form a fairly rigid structure. And what we'll have to do, there are special enzymes in here that will untangle these. And then we have to resort these proteins, putting them into membrane that will go into vesicles and recycle that, and others, the proteins, will put into the plasma membrane. There's a whole machine in here that disentangles this mess and then sorts the proteins in the right spots. We don't have time for that. But I'll talk about it in 325 if you take that course. All right. Anything else? Yeah.
Does Snare stand for anything, or?
It does stand for something, and I never remember what it is. It's got this great long, it has to do with NSF attachment proteins. And NSF is N-ethylmaleimide sensitive factor. And N-ethylmaleimide is a reducing agent. And it was first found to screw up any number of these processes. So they first found the chemical, and then they started naming things for the chemical, and it's lost all meaning at this point. But we still keep the names. Anything else? Okay, if not, let's now add in the next character. This is all well and good for releasing the neurotransmitter, but what we want to think about is what's going on the postsynaptic side. So here we have the postsynaptic cell. This, of course, in here, between the blue presynaptic membrane and the postsynaptic membrane, is the synaptic cleft. Let's redraw this. And bring together all the players in this story. So what we're going to find is that we have used the voltage-gated calcium channels to permit the influx of calcium. That calcium then binds to the synaptotagmin. This binding of calcium to the synaptotagmin triggers the winding together of the alpha helical regions of the V snares and T snares, smushing the phospholipid bilayer of the vesicle into the phospholipid bilayer of the plasma membrane. But what we want to think about is what happens after the vesicle fuses with the plasma membrane and the 5,000 to at most 10,000 molecules of neurotransmitter that had been sequestered, contained in the synaptic vesicle, are released into the synaptic cleft. So here's the postsynaptic cell. Now, as transmembrane proteins here in the postsynaptic cell, I'm going to put some of these structures here in orange. And if we were to talk about this molecule at the neuromuscular synapse, where Langley was working, this molecule would be a nicotinic acetylcholine receptor. But if this is in the central nervous system, and that's what I'll draw here, if this is meant to represent a central nervous system synapse, then these structures are most likely going to be glutamate receptors. Glutamate is the most common of all the neurotransmitters in the central nervous system. So if you're looking at a synapse and you're wondering what kind of receptors there are, it's most likely they're going to be a variant of the glutamate receptors. The glutamate receptor is a type of ligand-gated ionotropic channel. Well, there's meaning in that terminology. It's long-winded, but a ligand is something that binds or ties. So a way of thinking about this is the neurotransmitter. the neurotransmitter binds. It's a bit of a stretch. Binds or ties to the neurotransmitter receptor. Now, these orange structures, which are the so-called ligand-gated ionotropic channels, these obey many of the rules that we talked about for hormones. That is, on the on the extracellular side here, in the extracellular region out here, there needs to be a binding site that has a 3D specificity. A 3D specificity for the neurotransmitter. So it's a lock and key arrangement. The neurotransmitter needs to fit into the lock and key kind of three-dimensional structure here of the receptor. When the neurotransmitter, in almost all cases it requires two molecules of neurotransmitter, when the neurotransmitter binds to the receptor, that causes a structural change down in the middle of the channel. It swings the channel open. It basically increases the cross-sectional area of this part of the protein that's embedded in the lipid bilayer. When you increase the cross-sectional area, sodium, which we all know is in concentrations of roughly 130 to 140 millimolar in the extracellular space, that sodium now comes rushing in through the channel and comes into the postsynaptic, comes across the postsynaptic membrane into the cytoplasm. Now, an important thing here to recognize is that the neurotransmitter binds to the receptor to open the channel. It's a ligand-gated channel because we have to bind the neurotransmitter to the receptor, but it's ionotropic because it allows ions to come through. Many people will look at this and they'll think the neurotransmitter is coming into the cytoplasm. It's not. The transmitter never comes into the cytoplasm. The transmitter is way too big to get into the cytoplasm. But what the transmitter does is it triggers the opening of the channel. When it triggers the opening of the channel, we now allow for ions to come in. And remember, our rule is that when ions come in, they change the membrane potential. So this is going to be sodium influx to depolarize the postsynaptic membrane. The depolarization produced by the release of the contents of one vesicle is called an excitatory postsynaptic potential. So any questions to hear? Go ahead, you can't help yourself. So the question, a good question, what's the difference between this and a generated potential? A generated potential is what happens when we mechanically open those stretch-activated channels. This is a very similar event, but rather than opening stretch-activated channels, we're now opening channels to ions through the use of neurotransmitter. But you're absolutely right when you look at this. The more of these channels we open, the greater the depolarization will be. So these potentials will be graded with the number of ion channels that open. It's just the real difference here is a generated potential is a mechanical change, whereas this is a chemically induced change. How do these close? Oh, man. All right. These channels will close pretty quickly after they open. It's called desensitization. And what will happen is they open, they desensitize, and then we wait for a period of time for the neurotransmitter to come off. The transmitter tends to come off asynchronously, and so as long as one of the two sites is bound to a neurotransmitter, it remains in the activated state. But if there are, in this case, the central nervous system, our old friends, the glial cells in the neighborhood, they're going to take up the neurotransmitter, lower the concentration, so that these channels don't have enough neurotransmitter to open them. Okay. Yes?
Are there glutamate receptors that allow potassium ions?
Okay. I don't want to tell this lie. I have emphasized the point that sodium comes in. The truth of the matter is these channels are generalized cation channels, that they allow sodium in and potassium out. But under almost all circumstances, remember that the resting membrane potential is going to be down around minus 70 millivolts. It's going to be within a few millivolts of EK. That means there just isn't much driving force on the potassium to leave compared to the driving force for the sodium to come in. So under these circumstances, this is excitatory. Okay, but you're absolutely right. They will allow both sodium and potassium to flow. But let's not get into those weeds too deeply. Yes?
So it stands to reason that if there was an excitatory postsynaptic potential, there should also be an inhibitory one too?
That's for next time.
Okay.
All right.
But would that be caused by like hyperpolarization?
So the question here is, if this is a depolarization causing an excitatory postsynaptic potential, is an inhibitory postsynaptic potential caused by hyperpolarization? In many cases, yes, but let's hold off on that, and I'll give you the subtleties of that on Wednesday. Right. So what I want to do here is I want to superimpose this on one of our pineapple neurons. So let's do this here. So here's... So remember, this is input. And the hallmark of the input region is that it's enriched. It's enriched for ligand-gated, ionotropic channels. So that all over these out here will be hundreds, perhaps thousands of these output regions. So what I've drawn here with these squares is simply what we have in the upper board, trying to delineate the sites where the vesicles will fuse the plasma membrane. If we were to record from one of these sites, if we had an electrode in here, and we record, we would see the resting membrane potentials about minus 70 millivolts. And then at this point, we open the ligand-gated channels. In other words, right at this site, let me put another one of these blue ones in. Right at this site, we have the release of neurotransmitter. And in this thought experiment, we're measuring the potential generated across the membrane by the influx of the sodium. Let me leave this one up. We're measuring the influx of sodium at the site produced by the opening of those ligand-gated channels. And when we do that, we see the depolarization may go from minus 70 to maybe minus 69 millivolts. Another way of saying that is the few hundred to maybe a couple thousand of receptor molecules at that neurotransmitter release site. The opening of all of those guys only depolarizes the membrane from about minus 70 to minus 69 millivolts. Now remember, wherever we look at a membrane, we have these potassium leak channels. So now from what we've said from our model neurons, what's going to happen to the positive charge that's generated in millivolt depolarization as it propagates on this membrane? So it gets smaller and smaller. Now remember, out here, this is intended to represent the conductal region. And the hallmark of the conductal region is voltage-gated channels. So if you bear with me for just a moment. Here at this site, right next to the input region, is a region of membrane called the trigger zone. And it's at the trigger zone we cluster voltage-gated channels. Now remember, if we were to take this recording here, it goes from minus 70 millivolts to minus, maybe 69.5 millivolts, because the potential's gotten smaller. So we've established the membrane potential produced here is going to get smaller when it gets to the site. What I want to leave you with, so remember what we're trying to do here is bring the membrane potential to about minus 55 millivolts to reach threshold. And what we'll start on next time is how these tiny potentials have to work in concert with maybe a few hundred or at least tens of other of these potentials to add together or summate at the trigger zone to bring the membrane potential to threshold. With that, I'll let you go.